Hi, Ellen. This is Lindsay Womack. I am stepping in for Michelle today as she is out of town.
Okay, thank you so much
Yeah,
for filling in. And our
Sure.
presenter, it looks like he's here too. Michael.
Yes, I'm here. How are you guys today?
Good. So if you
And
have any issues, we'll have Lindsay here to help us.
okay, can I see if I can share my screen already?
Yeah, that'd be great. I got on a
Yeah,
little
let me
early
see.
just because I wasn't sure too.
Yes, I think I am sharing, correct?
Oh yeah, you did good. I can see your slide.
Okay, all right. I will wait for everybody getting in, then I
Yeah.
share again.
Perfect. Oh, somebody's coming on now.
So, Ellen, I'm just going to go ahead and ask everyone in the chat to go ahead and put their institution and their rank
Perfect.
just
And
to
their
collect
name.
that information.
And their name, so we have that on file. Thank you, Lindsay, because we do need that.
Okay, great. I will.
So Michael, put yours in too.
Sorry?
Lindsay's asking everyone to put their name in, their institution.
Okay, I will do.
Yeah.
Hey, everybody. I think we're still waiting for people to arrive.
And Lindsay is in for Michelle today. And Lindsay's asked everyone to please put their name, institution, and rank, whether faculty, graduate student, undergraduate student, whatever. Community.
Anything else you want to add? We'd appreciate it. So while we're waiting to get started because I see people are still joining. Thanks, Ellen, for that reminder. And just, gosh, this is our first meeting after our July event. Our June event. And it was great. How many people attended the slam dunk? Can we see some hands? Well, for those of you who did, I think we all had a fabulous time, and it was a great event on many fronts, and lots of presenters, our rec scholars and others, and we were just so appreciative of everyone who was able to attend. That was really terrific. Let's see, we're still gathering. Donna's put her name in, and others can add. While we're still giving it, Michael, if that's okay, and Ben, we're just going to give it a few more minutes.
Okay, no problem.
Okay, let's see. People are starting to put their names in. Keep doing that, please, as we get started. One more minute, Michael, just one more, and then because I want to leave you plenty of time, right? But we just hit the magic number for me.
No, okay, I have 2 hours, correct?
Well, we could listen to you all night long here, but perhaps just in the interest of efficiency, we could
Okay,
a little less.
I will do my best.
I have no doubt. For those of you who were at the slam dunk, we have Lindsay's got a hand raised. Lindsay, one more.
Oh, sorry. I was just raising my hand to say that I was at the slam dunk.
Oh, good. Good. Well, what did you think?
Oh, I loved it. It was great.
Good. Good. I think everybody had a great time and learned a lot. I know that I did, for sure. Okay. Well, with that, and people keep putting your names in the chat and your rank in the school would be great. And while that's happening, I'm just going to go ahead and turn the program over to our guest speaker. And I wasn't sure, Ben, if you were going to say a few words before Michael talked.
I'm happy to introduce today's Ideas Forum speaker, Michael Almeida. He joined my lab at UNC Pembroke at 2016, mostly as a volunteer. I saw how quickly he learned techniques, so I quickly hired him as a lab manager. And fortunately, he discovered a wonderful PhD program at UNC Wilmington, where he could join experts in the fields of natural products and cellular protection pathways. So I'm his major advisor because he does all his research at UNC Pembroke. We've had a few people doing PhD work at Pembroke, but Michael's the first one to do his entire thesis project on the campus, and it's a nice connection between the two UNC campuses. So take it away, Michael.
All right, thank you so much, Dr. Bar, for the kind introduction. I always say Dr. Bar tell more than what I am, but anyway, that's me. And it's a great pleasure to be here today again. And I was at Slam Dunk, was a great time. I put one slide, my last slide, I'm gonna mention a little bit about that. And also thank you all to accept me as a speaker today to share a little bit about my thesis work. It's almost like my pre-defense. Today, my pre-PhD defense, and I have my disclosure here, and I hope you all enjoyed that, a little bit what I have been done for the last 5 years. And I'm here, as I told you, to talk about my PhD work, that is the role of natural products to improve the cognition and promote brain health in general. And that, as Dr. Barr mentioned, is a dual campus PhD between UNC Pembroke and UNC Wilmington. And just a little bit about me. I am from Brazil, if I never told that before. I have a dual degree in kinesiology that is a little bit strange and out of the field. But I have a dual degree because I have a teaching kinesiology concentration. On that time, it was when I started research. I started researching clinical research over there to do some training to improve performance and runners. And that is clinical, not like a medical clinical research, it's more like a treat. And then when I finished, I started my master's degree in science, University of São Paulo. And during that time, I researched about how the physical exercise in rats can modulate the axonal transport in neuronal cells to avoid neurodegenerative disease. And then I trained the rats to run in a treadmill for a couple of weeks, and then I analyzed the changes in the brain and also did some cell culture and others to analyze those axonal transport in neuronal cells in a neurodegenerative field. I also have a few publications about that. And when I finished my master's degree, I moved to US in December 2015. It's looks like a long time ago, but as Dr. Barr told you, I enjoy Barr's lab as a volunteer. First day when I was here, I asked him, "Oh, can I be a volunteer?" It's because my wife was his postdoc. She was in postdoc training here. That's the reason why I met Dr. Barr here. And then he said, "Yes, you can join my lab as a volunteer, but I don't want any PhD students right now." And then he got tired of me. He said that I will not leave his lab. And then in 2017, he hired me as a research specialist. And then I turned as a lab manager too. And then in 2019, I put him against the wall and I said, "Dr. Barr, I'm here for my PhD, or I will leave for PhD, or I do my PhD with you." And then he got scared. And then he accepted me to be my mentor. And then I got inside the program to do my PhD in Wilmington, where I am a student there, but I developed my thesis here at Pembroke. Okay, I'm not here to talk about me, just a little background. I'm here to talk about my thesis work. And what I'm gonna cover today is, I divide that in 4 chapters because it's almost as my thesis is working on. And the first one is to talk about how aging affected the cognition. I have some perspectives on that, the cause, and also a potential solution for that. Then I will move on for an emerging field that is natural products that can be used to promote brain health. Then I will see from that natural products perspective, discover in vitro, if I can translate some of that results to in vivo. And later on, I will talk a little bit about how we can identify the fingerprint of these natural products that might promote brain health and cognition. Anyway, okay, but to start to talk about that, I always say that I have a good news before a bad news. And my good news here is we are living longer than ever before. And that is true. We can see that through the graphic here. And that, I think, is not a surprise for anyone. But what I'm highlighting that is because the aging is what I'm interested in. But what is aging? Aging is a biological and natural process. And aging is not a disease. And I use yet, because it's not a disease yet. You'll be a disease probably one day. I don't want to get in that discussion, but why is not a disease yet, and what's the relation aging has with disease? It is, aging causes a change in the whole body. We have a change in energy, we have a change in metabolism, we have a change in bones, and for some reason, I want to focus today in cognition. We have a change in cognition. And while aging is not a disease, it is well known that age increases the risk for a few diseases. Normally, they are a group of chronic disease or disease that's happening, has a slowly progressive, like heart disease, diabetes, or Alzheimer's disease. But again, that is the bad news of the aging. But the good news is science is doing a great job. And if you see the numbers of death for the last 20 years, what we have is among those most common aging disease, almost all them decrease or do not change, do not increase the number of deaths. Again, compare the last 20 years. But only one increase, that is Alzheimer's disease. And that is what I'm trying to discuss a little bit with you all today. What is Alzheimer? Alzheimer is just the most common type of dementia. Dementia is a big umbrella of disease, and understand that as a cognition problem that is going to affect your daily life. And again, Alzheimer is the most common. But why I need to mention about aging cognition that can lead to Alzheimer? That is because we have a race against time when we think about cognition. And as we age, we can have different levels of cognitive decline. It's true that I feel that they don't have that we call super aging. But normally, all of us are going to have this cognitive decline in different levels. We can have a normal aging, or can have a cognitive decline that can lead us to have a dementia like Alzheimer's disease, that comes like other proteinopathies and others age-related dementias or age-related disease that we can have late in life. Okay, but why we need to understand, or what we need to understand about aging that comes with that cognitive decline? To talk about that, we need to understand a little bit about the nervous system. And there is where everything gets complicated. Because for more than 100 years, we have been trying to understand the brain and the nervous system at all. And that is very hard. But we learn one very important lesson. That is, the major function of our nervous system is communication. What we need is one cell communicate with the neighbor cell. It's called synapse connection. And as much synapse connection we have, probably is a better cognition we're going to have. That is the main lesson that we have been learning for these last 100 years. But the brain is a high-tech machine. And this image here is just a short version of one map of the full abilities that, or full task that our brain can do. That is developed by an aerospace engineer. And if you want to just go on that website in the down left, then you can see the full map of that. But why am I put that here? Just to show you all the complexity of this network that we have in our brain to make it a high-tech machine. Okay, you might not believe on that. I will give you one example. That is a quote from the same website. If you take the same mass from a Cortex and compare if iPhone 8, that same max, we talk about 1 cubic millimeter here, we're going to have about 50,000 neurons. Each one can make 6,000 connections. That is the communication with a neighbor cell, and going to have over 3 million of connections or interconnections. If you compare the same space or the same cubic space for iPhone 8, we're going to have 23 million of connections. That is like our brain mass has 10 times more technology and more connection, or synaptic density to be more specific, than one A8. That is just to exemplify how high-tech is our brain. Okay, what is my work today? Part of that is try to simplify that to all understand what is my thesis work. To do that, let me just give a few numbers about brain. Brain have more than hundreds of billions of cells. We don't have just one type of cells. We have neuronal cells, the most famous one, and we have the glial system that is like a support cells. Those cells, as we know, make communication with the neighbor cell, and that is the main function of the brain. And how this happen? Normally we have a presynaptic cell, and then we have a postsynaptic cell, and the message is passed through one cell to the next cell, and then that's go as a sequence. You go in many directions. Remember about the complexity, but that is what our brain should do in normal conditions. Okay, another image that can represent that is this one, where you have one cell body, soma of a neuronal cell. I have the ramifications, and I have few spine heads. Each one of these spine heads can make a connection with the neighbor cell, can make a synapse. Okay, why all this is important? because it's simple like that. More synapses, we have a better neuronal communication. If you think about cognition like memory, you're going to have a better memory. And the opposite is also true. And that is what's happened during the aging process. As we age, remember about cognitive decline, we have a synaptic decline. In the now, that is really important. The best correlated marker with cognitive decline is synaptic decline. Even though we cannot track this very well in humans, that is proven science, the best correlated marker with cognitive decline is synaptic decline. And what's happened when we have dementia, like Alzheimer's disease? That synaptic decline that normally occurs, if that normal aging, you'll be accelerated. If you have lower number of synapses, you're gonna have a lower communication, probably gonna affect some cognition as memory, for example. Okay, one more image to try to exemplify how this affect our brain and can lead us to have like Alzheimer's disease type stress later in life. If I be able to separate one single cell from this universe of hundreds and billions of neuronal cells in our brain. We have here the cell body, we have the ramification, we have the presynaptic terminal, and we have for the neighbor cell, a postsynaptic terminal. As we age, we start to accumulate toxic forms of protein. We learn a lot about that during slam dunk time. And that toxic form of protein can appear inside and outside the cell years before we have symptoms. And that is going to affect the axonal communication and the intercell communication as well. And then if you affect these synapses, we're going to have problems in memory or problems in cognition. Okay, but our cell has an organelle called lysosome that is responsible to clean this garbage that accumulates during life events. But why then? When do we age, we are not able to clean up that, and why we accumulate is a bad news, but it's true. As we age, we also have age-related lysosomal decline. And then we have a synaptic decline associated with a lysosomal decline, and both are accelerated when we have a dementia type of cognitive problem or Alzheimer's disease, for example. Okay, if all that said, we have a hypothesis. That is, if we can modulate the lysosome, and remember, I call lysosome as a garbage disposal. If you can modulate this garbage disposal, the same one you have at the sink at your home, by modulate the blade that is inside, that for the cell is going to be the lysosomal protease that are enzymes responsible to break down protein, like cathepsin B, cathepsin D, cathepsin L, and many others, we might be able to clean this garbage, okay? But to understand that, we need to understand the macro process, that is autophagy or autophagy. I will die without learn which one is the right one, but I will keep, you guys are gonna understand me. This is a fundamental process that cell has to clean, unfold, misfold, or old or unfunctioned proteins, or even organelles like mitochondria, or even lysosomes. And are many types of autophagy. Are microautophagy, chaperone-mediated microautophagy, and others. But why I'm highlighting that, I don't want to get in details about each one. I just want to get one detail. Don't matter which type of autophagy we are talking about. I can include here mitophagy. Those autophagy are gonna share one content, that is the lysosome organelle. And wow, that is why it's important to modulate and to think and to target lysosome, because lysosome is autophagy as well, is a lysosome dependent mechanism. We cannot have autophagy without a lysosome, or not a functional autophagy without lysosome. And what is a lysosome? Lysosome is a double membrane organelle that is inside is rich in enzyme, and there is acidic field. That is why it's good for enzyme like protease to break down protein. And when you have an autophagy vacuum filled with that material that need to be degraded, Later on this autophagy process, we're gonna have a fusion of this vacuole with the lysosome. And then lysosome is gonna magically release all those enzymes in a single vacuole. And then those enzymes are gonna be responsible to break down the product that is inside that bag. Let's think like that. It's not only lysosome or autophagy responsible for recycling. We have another system like ubiquitin proteasome system that is a kind of work in parallel with that, but the measure to degrade this type of protein accumulation stress is through lysosome and through those enzymes. Okay, but it's feasible to target lysosome to improve the degradation and clean the cells? Okay, to do that, I will show few results from our lab and from others. You can see a couple papers on my right side here. And this one is one model of a mice model for Alzheimer's disease, a transgenic model. And we can see on this bar here in green that we have a high level of this form tox of amyloid beta 42. When we positive modulate this lysosomal protease called cathepsin B, we have a lower level of this toxic form. What means we have a degradation. We can clean up this toxic form. Same type of results we have for another protein accumulation disease, that is Parkinson. But now we're talking about another protein aggregation. It's about alpha-synuclein. And when you have this accumulation of alpha-synuclein, and we also modulate this lysosomal protease to work better, we have a degradation and decrease the level. We also have a mild cognitive impaired model that is like a pre-disease. I will talk a little bit more about that later, but it's like a pre-Alzheimer condition. It's not a disease, but came before and you have a worse cognition than normal aging. And then we have the same results. We don't have aggregation, then we don't have to worry or to think about degradation here. But when we have modulation of this lysosomal protease, we have enhancement on cognition of those rats model of MCI. Okay, another evidence that can tell me I can maybe modulate this lysosomal protease and improve memory came through physical exercise. Everyone knows here, physical exercise, and remember I'm kinesiologist, is good for health and good for the brain. And recently, a group from NIA, National Institute of Age from the United States, in collaboration with a German group, they published a paper where the outcome is cathepsin B released during physical exercise by muscle cells may be the mediator of the cognition improvement after physical exercise. They found that in humans and primates, and they confirmed that by having knockout mice for cathepsin B, where in absence of cathepsin B, the physical exercise don't have the same effects on neurogenesis, neuroplasticity, and cognition. Then again, is another evidence of modulate lysosomal protease can be a good way to avoid this type of cognitive decline. Okay, in summary, for my chapter 1, I talk about age can cause age-related disease like dementias and Alzheimer. That can be because synaptic and autophagy problems, and I show you guys we might have one potential modulation or one potential solution for that. It's important to understand that I mean not talk this is gonna save the aging. No, the hallmarkers of aging go over and have more aspect than that's it. I'm just touching laws of proteostase, like the cycling of protein and the cell-to-cell communication, the synapse. We might touch a little bit in nutrient sensing because lysosome do that job. And right now, Dr. Z is here. We are starting a new collaboration with ECU to understand a little bit more in our model about mitochondrial dysfunction. And then you're gonna just englobe more hallmarkers of aging in our research. But when I was preparing my talk, I saw one paper from last year. And remember, we have hallmarkers of age, and then you have change in the whole body. And one of the most important scientists right now, Ana Maria Cuervo, please remember that name. You guys are gonna hear some more about. She just enjoyed the American Academy of Arts and Science. She's a new member for this year. She published a review on autophagy and hallmarkers of aging. And the outcome from that review is that image here. In such a way, the autophagy process, that is a lysosome dependent mechanism, has some regulation in all other hallmarkers of the aging process. That is another evidence how important and why autophagy and lysosomal modulation might be one center or one key point to avoid cognitive impairment during the aging process. Okay. All these of my work give me a couple publications. I have a chapter that we published last year and a couple research papers about that. And then I moved to my chapter 2 of my thesis. Remember when I talked about we are living longer than ever before, and we're gonna have a high number of elderly people by the end of the century that we never have before. And the high change of that have a high number of elderly people is going to happen in poor countries. Why am I mentioning of that? Because those elderly, they're going to face different problems that we are not facing here in the US, like poor nutrition. And why I want to mention poor nutrition on dementia? It's because it's well known that a poor nutrition can increase the risk for dementia. As well, it's well known that a healthy diet can prevent against some types of dementia. Okay, you might not be convinced about that. If you just do a quick search, you're gonna find in everywhere a lot of evidence that natural products may be Alzheimer's disease therapeutic option. I put one here that you guys might remember last year in the opening of the ADRC at Duke-UNC. One of the keynote speakers was Rodolphe Tanzi. At the same year, almost the same day when he gave us a talk, he published that paper about one natural product called cat's cloth from Amazon forest that might be able to reduce plaques and tangles in a model of Alzheimer's disease. I just put others two papers here to highlight how much is growing that field of natural products and drug discovery. And the middle one is from Greensboro. And if you do or plan to do any type of natural products drug discovery, you need to read those work from Greensboro because they do a great work on that field. But okay, back to my work. As I show in Slam Dunk, my work is tied to find natural products that can work to modulate this autophagy, promote synaptic integrity, and avoid cognitive aging or cognitive problems that comes with aging. Okay. In other words, or in simple words, what I'm looking for is how I can improve synaptic integrity and lysosomal modulation or associated with lysosomal modulation to avoid cognitive aging. How I did that? I use a model of mature hippocampal slice culture. This model is also called organotypical brain slice culture. This is a well-established model, and that the main advantage to use that is because it's a long-term culture. What is really important for the aging process is a mature brain characteristic. If you see those images here that I'm highlighting here and see, we can see after a couple of days, and after the maturation process, we have the same synaptic density and glial densities as a mature brain. And then we have multineuronal cell type. As I told you, we have neuronal and glial cell type. This is a tissue, is not a monolayer cell culture. Then we have cells in multi-dimensions, what is pretty important for cell-to-cell communication because kind of represent what we have in our brain. And that is well established as a translational research or easy model to do translational research. Okay. Back to my study design. I have this organotypical slice culture. I treated them with different natural products. Here I just listed the most common one, like Asian ginseng, white blueberry, bacopa, green tea, pineapple, and others that I'm not listing here. And after 3 days, I did a harvest it and I did some biomolecular analysis. Okay, what is the first question that I'm trying to answer? Does these dietary supplements act on lysosomal protease? For that, I measured the level of this lysosomal protease called cathepsin B, and I found among a group of natural products, 4 of them increase the active form of cathepsin B. Then I look if those increase some synaptic markers, and I look for GLU-A1. GLU-A1 is a post-synaptic marker called glutamate receptor unit 1, and that is more than or 90% or more of the synapses in the hippocampus is a glutamatergic synapses. And then we found among those 4, only 2 was able to increase cathepsin B and also the levels of GLU-A1. Okay, we did some correlation analysis, and then we find that only American ginseng has a positive correlation of this increase of synaptic integrity markers and active form of this lysosomal protease. Okay, what we did after? I want to see now if after I select these natural products that improve synaptic integrity associated with lysosomal modulation, how they will respond in an aging model. To represent an aging model, we try to represent this proteinopathy that we have during the age, that is lysosomal dysfunction. To do that, we use a drug called chloroquine. Chloroquine is a drug that is internalized by lysosome, and when this is inside lysosome, it changes the pH, and then it's not acidic anymore. The enzyme is not work properly. We're not going to have degradation. Then chloroquine is a lysosomal inhibitor, a kind of the same disturbance that we have during the aging process. Okay, how I did that? I have the same organotypic slice culture. I pre-treated that with the selected plant extracts for 3 days. Then I co-treated with lysosomal inhibitor in absence or presence of natural products, and I did some biocellular analysis. Okay, as expected, I know lysosomal inhibition causes a reduction, a significant reduction in the levels of these synaptic integrity markers. But in presence of this American ginseng, we kind of preserve the synaptic decline on this mature brain tissue. Okay, I did some others analysis like confocal analysis to see in red post-synaptic marker and in green pre-synaptic marker, and I have the same results. Presence of American ginseng, even in presence of lysosomal inhibition, preserve the synaptic decline that normally occurs during the aging process. Okay, let me give you another evidence that American ginseng can modulate cathepsin B. In 2016, this paper came up in a Nature group about cathepsin B mediated the cleavage of a protein called Disable2, DAB2, to be friend with us. This is one of the figures from that paper. We can see DAB2 in green. We can see cathepsin B in red. How they found that, and this is a good example to we understand. As cathepsin B increase, and then we can see this across the day in these middle panels. Remember, cathepsin B is the red labeling. We have a decrease on that DAB2. They have a breakdown of DAB2. And remember, cathepsin B mediate the cleavage of DAB2 protein. Okay, what I did, I test what's happened with DAB2 with American ginseng. And then I found when I have American ginseng, even in the presence of lysosomal inhibition, I have a high level expression of this breakdown product from DAB2. We can see on this graph, the breakdown products about 66 kilodaltons is elevated in presence of American ginseng. Okay, another evidence that American ginseng can improve the autophagy-lysosomal pathway can be related with the diagram here. On this, during the process of autophagy, we have different markers, and I look for the marker called P62, that is a kind of autophagy receptor, and also I look for LC32, that is a kind of marker of late autophagy. And then what I found, American ginseng have positive effects on that, protects or induce autophagy flux to change the levels of these two markers. Normally, P62 is elevated when we have a problem, and LC32 is also elevated. Okay, let me give you another evidence before I get more details about that. That is the table here. We can use the rate between LC32 and LC31, that is, LC32 is a late marker of autophagy, to measure the autophagy flux. It's an indirect measure. How this work? We need to have some inhibitor of the autophagy process, and that's gonna increase the level of LC32, but that indicates a stress. If you have a drug or a non-pharmacological intervention that increase the level of this rate over the inhibition rates, that means you have activation of autophagy flux. That is what we have here. When we have this American ginseng, this number is higher than the inhibition. And together, DEB2 breakdown product plus P62 modulation plus LC32 by LC31 increase in duration, I have enough evidence to show, yes, American ginseng appears to modulate the autophagy-Lysosome pathway. Okay, that is how I finish my chapter 2. This work, we are work to submit that is almost done. This is, I hope, it go pretty soon. And that's moved me to the chapter 4. That is, can I translate that for an in vivo model? And here is where I talk a little bit about mild cognitive impairment. Again, mild cognitive impairment is not a disease, it's a cognitive dysfunction that we have between normal age and a dementia level of cognitive dysfunction. And remember, that is between, or happens before the Alzheimer's disease occur. And to do that, we have a rat, is a female rat model of mild cognitive impairment. We prepare the food supplemented with that natural product. We supplement the rats and keep the control as need for 6 weeks. And then I did some cognitive assessment to see how this might or not change the cognition. And then I'm doing right now is the biomolecular analysis. Okay, let me see how much time I have. I still have 2 hours, yeah? And then, okay.
Well, Michael, we do want to save a little time to talk, right?
Yeah, sure, yeah.
Yeah.
I'm not gonna get details about the test. I will be pretty happy to answer any question to chat later about that, but we use some well-established cognitive tests to analyze learning and memory and all. And this first one is a shock box we can use to measure learn. And I go just to matter of time, I'm gonna just go to the results. The first outcome I have for that is the American ginseng protects the learning process on these middle-aged rats. As we can see here, green represent the young group. MCI is representing red, and blue represent MCI treated with American ginseng. The blue bar show us we are not good as young. We do not expect that, and I'm not thinking young is the best age, but for this test, but we are not get the same level of cognitive decline, in this case learning, as the MCI. Is a short evidence that yes, American ginseng might protect a little bit this cognitive decline. Another one is when we have this shock induced learning. We can see after shock, even the control reduce the time spent in the dark. But we have a greater reduction when we feed or supplement those rats with American ginseng. Okay, I have others evidence or others analysis statistics to show that yes, shock induced learn and memory on these rats because they expend low time or short period of time after shock in the dark, where should be the shock, but if they remember, they will avoid the dark side. If they do not remember, they will not avoid. Okay, another test that we did is about exploratory habituation. Rats are explorers. And, oh, sorry. This video here is not a rat, it's a mice from another study, but just represents how we do that. We put the rat in a maze box, and then they cannot see those lanes, but we just track them during time where they walk and for how long. And then you can track that for a period of time. What did it give as a data to us? We compare first and second day and add that image represent. That's a real track for one of those mice. Again, that's not the rats. The day one, they walk much more than day two. And what is the outcome from that test for us? Imagine if tomorrow they decide to open a new mall center at Raleigh area. The first time when we're gonna visit there, we might walk everywhere as the rats did in the day one. But on the following day, when I go there again, I say, oh, you know why? On that side has a couple of stores that I have no interest. If I remember, then I not go there. That is exactly what we are measuring here. It's exploratory habituation that give us a little bit or an outcome about memory, a short-term memory. Okay, to present this data, the first thing I need to show is no difference in mobility or other marks that might affect the exploratory habituation of this rat. And as the previous results, American ginseng appears to decrease the exploratory habituation the second time, what may be one indicative that American ginseng induces memory or protects the memory against the aging process on this model. Another couple evidence that we have, we did some tests to analyze the anxiety and the exploratory for a single day of a new area. And those rats supplemented with American ginsengs appear to be low level of anxiety and high level of exploring a new environmental. And we did some different statistical tests to prove that. And also we did other tests like NATLET scores to see the level of stress or hang or flip test, where we kind of have the same results. Yes, American ginseng appears to protect or to delay these Asian cognitive change. Okay, that is my chapter 3. I'm almost done, I promise. Then when I finish, or I'm not finished this yet, that's not the true, but during my work, we figured out that we have a caveat in our research, or better than a caveat, we have a point where we can try to understand what is the neuroprotective properties of these natural products. And then we try to do some metabolomics and proteomics of that. The first one is in collaboration with UNC Wilmington, Marbionic Group, called DISCO. We are trying to see from that natural products that we screen and we find them can promote synaptic integrity, we try to identify inside that the neuroprotector compound. Let me give you one example about that. I have two types of ginseng, for example. We have Asian ginseng and American ginseng, and I have no geographical choice on this here. This is only American ginseng has a positive effect for the question that we ask. And then if they are similar in composition, we might run some mass spectrometry and liquid chromatography and do some target and or untarget metabolomics study, and we might be able to identify the difference between them. And then if we'll be able to test those regions on a biofragmented study, we might identify where is the neuroprotective aspect. We can see here, I have this region here on American ginseng that I don't have on Asian ginseng. And as well, I have this one here that I have in Asian ginseng, but I don't have in American ginseng. They are quite the same product, but they are not equal. And what is the outcome from that? I might never found, I do not believe, by the way, that I will find the neuroprotectants, because if you study natural products, you're gonna see it's not a single compound. They might be act together in different aspects, synergists, agonists, antagonists, to help one each other to be neuroprotective. But we might be able to understand the regions or compositions of these compounds that might help us to develop new drugs or new products that can help against this type of cognitive decline. Okay, next, again, this is a collaboration with NC Wilmington with Dr. Strangeman and Dr. Wilson. And next one is start because slam dunk. I put it here to not forget to mention that during the networking time on the slam dunk, we have a conversation in our table about proteomics. And then we decide, why not test proteomics in our model? And we are just start. We don't have anything else besides this slide here about this, but the idea is here. With a collaboration with NC State, the metric group, we are trying to analyze based on cognitive performance, brain samples to do label-free proteomics to identify what natural products change in the level of proteomics on those brain where we do have protection and also where we don't have protection that can be used as a biomarker. As well, we might do some target proteomics to identify autophagy-lysosomal or synaptic integrity related proteins that also can be used as a target or biomarkers for this cognitive follow-up during the aging process. Okay, if all that said, I would like again to say thank you one more time to have me today here. It's a great pleasure to share my research with you all. I would like to say thank you for half of my brain that is in Kinsey Lab at UNC Wilmington and half of my brain that is at Pembroke with Bar Lab. Thank you for all the agents that indirectly or directly fund this project. I would like to mention a little bit about Bioimage North America. If you not join that. I am part of the work group for development microscope for underrepresented students there. And then if you're not, please, it's free, just join us to help us to do that work. And again, you might all remember, please remember how we say thank you in Portuguese. Don't tell me that you guys need American ginseng for the next time. Obrigado, and I'll be happy to answer any question.
Obrigado, Michael.
Oh, thank you so much. You see, you don't need American ginseng right away.
We can all learn, hopefully. So thank you for that very exciting work. Let's see if anybody would like to ask a question or make a comment. Out there, I'm looking at their chart here. Change my view to see the gallery. Okay, and that was a request on the abstracts about, Ellen had asked, did you do your research with the American ginseng root or capsules and so forth? And Ben had replied, well, Michael, was still on the slides, that all natural products were prepared abstracts. Do you want to say anything more about that, Ben? About the type of extracts that were used? Or Michael?
Well, I can just say that before Michael joined my lab, I was approached by a company called USANA Health Sciences, and it was rather blind. They just sent us some extracts that they use to do combination supplements. So it was a blind study as far as what we were told of which ones they owned or which ones they thought were the better ones. So that's the work we're doing, and they haven't been involved in the manuscript process.
Great.
And they're just interested in what we find.
Lovely. Thank you. Ersped has her hand raised next.
To unmute myself. I keep forgetting to unmute. Michael, this was such a cool talk. Brought back memories from Max Planck, where we were doing a passive avoidance test. So I go back a little bit to that test. It's dependent on CAM kinase 2, right? Did you look at CAM kinase 2 phosphorylation in the brain of these mice?
You ask me if you can look for phosphorylation of what, sorry?
CAM kinase 2 alpha.
Yeah, we probably can look. And again, we probably has much more than just modulation of autophagy markers and synaptic markers on that.
Mm-hmm.
Probably do have a lot related with mitochondrial health and many others type of homeostatic process inside the neuron. I cannot tell you that you're gonna have a change or not because I do not test. But again, natural products, they can be combined by 100 to 1,000 of different compounds, and which one of them or combination of them can produce different results. And again, I cannot say yes or no for something that I not test. But again, we are always interested in do some collaborations and try to see if the others questions can be answered by these two.
Yeah, just run some Westerns from CAM kinase to alpha. Because it's
Wow.
a central memory for memory. So it would tell you that indeed this is a strong
Yeah.
proof at molecular level, you know.
Yeah, again, we can chat more a little bit about that to me get a little bit more what you're talking about. But again, we are happy to do any type of collaboration, try to decipher a little bit more the mechanism of action or how this happened.
Thank you.
Rachel, you have your hand up?
Yep. Hi, Michael. Nice to see you again and nice talk. I was just wondering if you thought potentially you would get bigger responses, particularly with looking at some of your cognitive measures and your behavioral measures, if you did a longer duration of treatment, so maybe 6 months or 3 months versus just the 6 weeks.
Wow, that's a great question because we think about prevent aging, not treated aging, and then that's a good question because if someone asks me what is the correct number to use, say that we're going to prevent wages 1 week, 1 year, or for the entire life, I don't know. If we follow up that exercise probably is the best way to prevent some type of disease, we're going to think we need a lifetime supplementation of that. How much that's going to be, we can follow up. I don't know for how long, but for sure, I think if we do a long-term study, we might gonna have a better effect on that. I don't know what's gonna be the minimum because we did not test, but I'm sure time is a very important dependent on that. And because of time in our side, not time of the rats, we cannot like test 1 week through 1 year. But it'll be a great question. And again, we need to think about prevent aging. And when you think about prevent aging, probably it's not going to be one magic pill that we're going to drink and we are young again. But it's something that we're going to use as an intervention, like a diet or a lifestyle that we change. That is something that we need to think about. But yeah, it's a great point to thinking about.
I can chime in to say my 13-year-old dog took heavy notes during this talk. She wants to know if it has anything to do with the age-related effects on eyes because she's completely blind and she's mad.
Great question.
Yeah, Dr. Bar asked that because he knows the field, and then Asian macular related disease, they have some problem with clean of garbage in that accumulate to is a kind of the same protein accumulation stress, not as the same as Alzheimer's disease, but they have a kind of this autophagy-Lysosomal problem. And then we might modulate. There are few trials that I try to use drugs, and Dr. Bar is aware about that few try to use drugs to try. And again, the importance is we are in the Alzheimer's field, but not necessary. That is just autophagy modulation is not benefit only for Alzheimer's. We try to have a collaboration. Dr. Bar and I has a collaboration a couple of years ago with a Nobel Prize from Chapel Hill that they study kidney disease and diabetes. And we just figured out in such a way that we can kind of modulate lysosome might be helpful to them too in another completely different aspect or different disease. You try dogs next time, Dr. Bar.
And for grad students or any researchers that are listening, it's picking the right model to do research is important, but also you got to convince your PhD committee that you're not pressured to get publications based on the model, because MCI is rather new and everybody's debating what is a good animal model of mild cognitive impairment. And Michael chose one that's in the literature, but they keep changing. So we don't know if the model animal and age we chose will be convincing to researchers, but it's middle-aged animals experiencing cognitive decline.
Do you think some of the abstract the extracts, natural products could have unintended consequences that are not positive?
Wow, that is another hard question to answer. To do that, we need to test, not guess. But one thing that I can do, those rats that we did, the supplementation, we save key organs that's normally used by pathologists to
Good.
do those analyses. And then in some point, we are just do that analysis to see if we have, like we do any other drug discover, if those supplements can cause any change, good or bad, to key organs like heart, kidney, and many others that we save. But we have those samples to be analyzed when is necessary.
So I think that's great. So as a clinician, I prescribe a lot of drugs and see a lot of supplements, and there generally is always some sort of side effect that was not intended or anticipated by the developers of the compound. That's why post-marketing research is so critical to drugs from the pharmaceutical company because of that. So I applaud you looking ahead to keep the ability to explore that, because that'll be critical, if this continues to be, positive in terms of your outcomes.
Yeah, correct. And we kind of work in collaboration with a group from UNC Chapel Hill on that side, because always when we try to test some drugs, we have a pathologist aware,
Excellent.
and she is who advises us, oh, that's what you need to save, that's what you need to do. And
Yeah.
that was we look at one day.
Great.
Yeah.
Other questions or comments for Michael or Ben? Just looking around. Well, it's just a couple more minutes to the hour, so not seeing any hands, which I might miss one, but if someone has an urgent question, just shout it out. But I just want to thank you again, Michael, for your great presentation, such exciting work, and Ben, for introducing us to Michael and your lab there that's doing such great work. And to all of you and the trainees and scholars, we're so excited that you're here and part of the forum, and we just want to continue along in our monthly presentations, and they can be presentations such as Michael's, or again, we can change the format should you so wish. And we're very excited. We have a number planned out already, but those of you who might be interested in speaking to the group or sharing your work or just bringing up controversies, that would be great with us. So just please let us know.
Right, and the next one is scheduled for August 25th. It's Daniel Parker. He was on, but he's not on right now. At 4 o'clock again. Thank you.
And everyone, please spread the word. This is a very nice avenue to practice your thesis defense for those graduate students getting that close. So I like the ideas forum is really helping many people.
So we're delighted to see you all, and we have 2 minutes to go here for any last comments. Be sure and make sure we have your name and your rank and your serial. No, not your serial. I just want your titles, or are you a student or a junior researcher or what? That just helps us. And so we'd appreciate it if you could add that information for Michelle. So thank you so much all for attending. Thanks to all who attended Slam Dunk, and we look forward to seeing you in a month.